Good morning, everyone. Apologize, we had some small technical glitches trying to get some things set up, but it looks like everyone made it into the room fine. We will wait a few more minutes to see if anyone shows up for the first session. As it is 8 a.m. though, I'm not sure how many people will show up, but we'll wait just in case. Can everyone see the presentation okay?
Yes.
All right, cool. All right, it's 8 o'clock, so we'll go ahead and get started. Thank you everyone for coming out and taking part in this year's Graduate Research Colloquium. My name is Nathan Ford, and I'll be your moderator for today. So just a couple of ground rules before we get started. Each contestant will have 12 minutes to present. Your time will start the second you start speaking. I'll give you a 1-minute warning by interrupting you. Once you are ready to present, give me a verbal cue like start, etc., and I will advance your slide for you to begin. During the presentation, give me some verbal cue like next, advanced, or proceed to advance through your slide deck. I'll be introducing you before you talk, so you can jump directly into your presentation so you can save on time. At the end of the presentations, judges can unmute themselves and ask questions to the participants for about 5 minutes. While the judges are scoring, I'll moderate questions from the general audience, and I ask the audience members to stay muted during the presentation and to drop any questions they may have in the chat box. So before we begin, does anyone have any questions? All right, then if not, we'll get right underway. Our first speaker is Karef Alafari with their presentation on an efficient computational model to study immiscible two-phase flow in porous media with validation to Berea sandstone. So whenever you are ready, please feel free to unmute yourself and let me know.
So who is going to move the slides? You are going to move?
Just let me know in some form or fashion when you want me to advance your slides.
Okay, yeah. Just I need to remove this, please. Yes, I am ready.
All right, you may
Hello.
begin.
Yeah, thank you. Hello, everyone. So this work is part of my research, which is titled as an efficient computational model to study immiscible two-phase flow in porous media with the validation to Berea sandstone. Next. So the motivation behind this study is the importance of immiscible displacement in many applications. So for example, here in oil recovery, so there is, they are injecting gas or liquid to produce oil, and there is a high amount of liquid oil is trapped in the porous media, and there is a high interest to produce these amounts. Also, there is another application that they have these issues with the trap saturation with the immiscible displacement. So the main objective of this study is to modify what is known by fractional flow theory equation by including the capillary effect to study the immiscible displacement and then estimating the trap saturation. Next. The presentation will be in three main parts, starting with the porous media probabilities and concepts. Next. Yeah, so basically porous media is a solid matrix with void space, which are the pores. The main property that we usually use to as a property of the porous media is the porosity, which is the void volume to porous media volume. Another important property is the permeability, the capacity of porous media to transmit the fluid. So permeability is unlike porosity. It's considered a dynamic property because it is estimated using Dart's equation, as we see in the middle, and by conducting a flow experiment. So it's considered a dynamic property and porosity considered a static property. So in case if we have a single phase flow, the figure below left, that's mean, and we are estimating the permeability, that's mean we have absolute permeability, which consider a property related to porous media. However, in case of two-phase flow, the permeability estimated or calculated is considered effective permeability because the permeability is not only a property related to the porous media, but because it is also related to the saturation of each phase. What I mean by the saturation means is the liquid volume occupied by the pores to the total volume pores. Next. So from this effective and absolute permeability, we will go through first important concept related to the fractional flow theory model, which is the relative permeability. So basically relative permeability is the ratio of the effective permeability to the absolute one. Relative permeability is a concept is used to specify the permeability of multi-phase flow in porous media. So the relative permeability can be written in terms of saturation. And there are many techniques to estimate the relative permeability. And the simplest way, which is, it's called the steady state method. So what they are injecting, in case of two-phase flow, they are injecting many different fractions of the two phases. And they take the advantage that the pressure drop will be same for both of them, and then estimating the saturation and the relative permeability based on that. For example, the figure below to the left, we can see the dot on each curve. These two dots, that saturation is just represent one test. So they need to cover the whole saturation, and then they get correlation for the relative permeability in terms of saturation. So this will be the very important concept that used for the fractional flow rate model. Next. So the other concept is the capillary pressure. Basically, capillary pressure is the combination effect of interfacial tension, wettability, and the geometry of the pore, all combined in the Laplace equation we see in the under the section of capillary pressure. The figure in the middle to the right is the general example that we use to see the capillary effect. Here I am showing two different pores at different sizes to show that the capillary effect is based on Laplace equation can be different in terms of the level of the fluid because the geometry or the pores are different. Also, in terms of the porous media, the figure in the middle, I am showing two-phase flow. The blue one is injected and the red one is displaced. So the concept of this capillary pressure can be seen in terms of the immiscible displacement in two main regimes. If we zoom into the pore level, so the figure to the left below, it's showing a pore and there is injected fluid which is wetting to the surface. We can see the liquid is like to spread on the surface of the pore. At the same time, the capillary pressure acting in the same direction. In this case, the immiscible displacement regime is called imbibition. The figure to the right is when the injected fluid is the non-wetting phase. We mean by non-wetting phase is the fluid doesn't like to spread on the surface of the pore. And the immiscible displacement regime is called the drainage. So these are the main two regimes that we can see in the porous media. Next. So now let's go to the concept of how can I use the capillary pressure in the fractional flow theory. So to the right, I am showing a very simple porous media. It contains three pores, starting from R3, the smallest pore, to the biggest pore, R1. So the figure beside to the left, I am showing that as we start injecting the red fluid, which is in this case imbibition, that's mean the injected fluid is the wetting phase, we will see the maximum capillary pressure because the radius is too small. And as we progress in the percolation, it's covering the whole pore, the smallest one that then it's go to the medium, then the largest. So we can see from this that two things. The first one that we can find a correlation for the capillary pressure in terms of the saturation. And the other thing is that when we injecting the fluid, we always the pores in case of imbibition or the wetting phase, it always go to the smaller pores until the end, it go to the largest pores, and the opposite in case of the non-wetting injected fluid, which is drainage. So that means that curvature will be same, and there is some hysteresis at the end. So from this, Leverett was able at 1940 to provide a correlation for the capillary pressure in terms of the saturation by providing the curvature of capillary pressure with the saturation as well to other properties like sigma, sigma surface tension or interfacial tension, and phi, which is the porosity, k is the permeability. With the curvature, he able to provide a correlation for capillary pressure in terms of saturation. And this is the main second important input that we are included in the fractional flow theory equation. Before going to the next section, there are two terms or constant that needed to understand, which are the capillary number in the middle of the slide. So the capillary number is is a constant represent the viscosity ratio to the capillary ratio, to the capillary effect. So the viscosity effect is the flow rate or the speed times the viscosity of the injected fluid to the surface tension. The other constant is the viscosity ratio, which is the viscosity of the injected to the displaced fluid. These constants usually used to characterize the immiscible displacement in porous media from the wetting area. We can see the figure right below that. We can see the half part below is there is the capillary force is the dominant, and we can see the fingers in the figure. And if we go up, if the capillary number increases, the dominant force will be viscous, and there is a different viscosity ratio. We have a different regime. Please, next. So now from these concepts, let's go to the fractional flow theory. So next. So fractional flow theory is a way to understand the immiscible displacement in porous media. So there are in case of two-phase flow. So basically, it's derived based on the flow rate of fraction, which is the first equation at the top, related which is the ratio of the injected of the phase flow rate to the total flow rate. And then the second equation is the general equation for Darcy. And then in our case, we are including the capillary pressure at the interface of the two immiscible fluid. We end with the general equation of the fractional flow rate theory. So it's contained three main parts. The first part is the viscous effect, then capillary, and then gravity effect. Next, please. So at the top, we can see they used to neglect both the capillary and a gravity effect, and they only consider the viscous effect. And in this case, the fractional flow rate equation will be in terms of saturation, since the relative permeability is in terms of saturations. So in our case, we neglect the capillary effect, the equation below, and then we just consider the viscous and capillary effect. However, if we see the term of the capillary effect, the capillary pressure will have the derivative for distance, and we don't have such correlation for the capillary pressure. We have correlation for capillary pressure in terms of saturation. So we simplify this term by using the chair rule. So partial PC over partial X will be equal to partial capillary pressure to saturation. We have a correlation for capillary pressure in terms of saturation. However, we need to deal with that other term, which is the spatial derivative of the saturation. We found that this term can be equivalent to -1 over ipsi, and ipsi is a length scale derived by Medici and Allen. So in this case, the term partial PC over partial S can be written at the end in terms of saturation, and the whole modified fractional flow rate equation can be written in terms of saturation.
You have 1 minute left.
Oh, next, please. I thought I have 12 minutes. Yeah, so there are some correlation will be used to this model, relative permeability, capillary pressure, other constant. Next, please. So the figure right, left is represent the deviation from the original solution, which is the blue one. As the capillary effect is very low, we have a high deviation, and then it's converged to the original solution. To the right is the derivative for the fractional flow rate equation. So, however, we consider the negative part from the right curve is not a solution, then it's a trap saturation. Figure below left is the trap saturation extrapolated based on trap saturation with the capillary number, which is consistent with the result of other experimental. Next, please. For the validation, so we can see we also use calibrate the model with the Berea sandstone. We have this result below, the figure below, which has a good approximation for the model provide a good approximation regarding trap saturation. Next, please. So this model.
That is 12 minutes. Your time is up.
Any questions?
Yeah, we can open the floor for questions from our judges.
I have a quick question. Could you didn't have time to say the conclusion. So could you please use one sentence to summarize your contribution for this?
Yeah, so actually the most important things of this model is to show the dominant forces in the immiscible displacement, and then specify the trap saturation.
Thank you. Another thing is that the picture you just showed, it's some experimental results, or it's some simulation.
These are, so the dots are experimental results, and the lines are my model result.
Okay, thank you.
You're welcome.
I have a two-part question, please. So I guess, could you repeat again, what is the main significance of this project, or how would it be used? And then after that, what are the specific tasks? Are there any experimental measurements that you're making? So what are the tasks that you are doing and that would finally ideally confirm your model or show its comparability to whatever is used right now?
So the main point of this model, it is a very simple model. We just need a correlation for the correlation and properties for that porous media. Then from these correlations and these properties, we can specify which dominant forces are dominant at a specific capillary number. And this is they are spending millions of money to understand and specifying these dominant forces, and also and how to specify the trap saturation. You can't imagine how much they are spending just to specify each dot of this experimental data. So these, so this model is very simple, and based on this correlation, and I can get any porous media based on these correlation for relative permeabilities and capillary pressure as well to the other properties, then I don't need these experiments. I can use this model to specify the dominant forces and then specifying the trap saturation based on the capillary number.
Okay.
Also, in the original solution, actually, they neglect the capillary number. So we will not see any trap saturation.
Let's see. Is there time for me to ask one additional question?
Oh, yeah, of course.
Okay, then I am under the impression that this would be used for something like fracking. If that is the case, then are there any measurements that people would need to do in the field based on the composition of, the solids that they're drilling through to apply this?
Yeah, so they need, as I mentioned, this model based on experimental correlation, which has the relative permeabilities and the capillary pressure. So we need this correlation. So this is also hard working to get these correlation, but this model will, the other point from this model, this correlation doesn't tell anything about which forces are dominant. It's estimated based on a steady state method where the capillary effect is neglected. So this model helped to understand that which dominant forces that we can find. And then from these correlation, we can get the trap saturation. Yeah, so we still need get these experimental correlation as an input to the model.
Okay, all right, thank you very much.
You're welcome.
Do you have any other questions from judges or audience members? All right, it does not look like it, so we'll give the judges a second to begin their scoring of the presentation. And if you guys just want to indicate that when you're done with a little thumbs up or yes in the chat or something, then we'll be good. But while we wait, we'll let Priyanka get situated.
Are we allowed to converse with the other judge to, I guess get feedback, or are our scores supposed to be completely independent?
I believe you can discuss. So if you need to send them a chat message, or if you guys want to stay in the room after both presentations are done.
Okay, I think that would work better for me if that's okay.
Yeah, that's fine.
Just a quick question. So do we need to input one score, or each judge can input their own score?
You should just be able to put in your own score. Let's see if we have the score sheet.
I would like to pitch in here. Each judge will have their own standalone scores that you can input through the Google form. But if you would like, you can discuss about it after the meeting, after the presentations.
Yeah.
Okay, got it. Thank you.
Do you have the Google form available?
Yes.
Okay, just making sure. Getting a thumbs up from Dr. Sung. Dr. Hayden looks like he's still scoring some stuff.
Sorry, I was just still thinking and looking at things, but yes, I'm perfectly fine with moving on.
Okay. Well then, next up we have Priyanka Kadev with her presentation, Capture and Release Care, a novel protein purification technique. So Priyanka, same as before, you'll have 12 minutes and that will start as soon as you tell me to advance to the next slide.
Hello everyone, and welcome to my presentation. Next. So in this presentation, I will be talking about a general introduction about proteins, some conventional protein purification techniques and their drawbacks, a new technique that we developed, which we named as capture and release to overcome some of these challenges. Next, I'll be talking about the validation that we did for care, and in the end, conclusion. Next. So the basic question, what are proteins? Proteins are the building blocks of life. Almost all of our biological processes that take place in our body, like signal transduction or cell growth, requires proteins. So the proteins are basically made up of the monomers called as these amino acids. And these amino acids, when they link together, they form a peptide chain, a polymer. These peptide chains further fold to form several conformations, which makes up the entire protein. So all the enzymes, storage proteins, receptors, transporters, hormones, defense proteins are all the examples of proteins. Next. Proteins are at the center of industrial and academic research. So most of the food industries, biotechnological industries, next, agriculture and pharmaceutical industries, all these industries require proteins. It is like a heart of the entire industry. But these proteins require, but these industries, all these industries require proteins in their purest form. Next. So in order to study the structure function relationship of a protein, it should be purified first. And therefore protein purification is a very important process, but at the same time, it is also a tedious process. Next. So I have mentioned some of the conventional protein purification techniques that are commonly used. One is the affinity chromatography that purifies protein based on the binding specificity of a protein. Ion exchange chromatography on the ionic properties. Size exclusion chromatography and electrophoresis separates protein based on their molecular sizes. So all these conventional protein purification techniques, which are mostly used, they are time consuming as well as they are expensive. So right now I'm going to explain about affinity chromatography. Affinity chromatography is one of the most widely used technique. It is like a backbone of the protein purification technique. This affinity chromatography I'm going to explain in terms of a protein called as galactin-3. The purple color is my galactin-3, the target protein. So in the first step, we load the crude sample, which is the entire mixture of sample, which also contains the target protein. And this particular target protein, purple, have the affinity to bind to the lactose agarose column. The yellow are the lactose agarose column. So in the second step, all the proteins, when they pass through the column, all the unwanted proteins or the other proteins in the mixture will not bind except the purple target protein, which has the affinity to bind to the lactose. Only that protein will bind to the column. In the third step, in the washing step, all the unwanted proteins are being washed off except galactin-3, the purple one, stays bound to the column. The fourth step involves the addition of free lactose in a very high concentration such that this particular lactose will elute out galactin-3, the target protein, the purple one, out of the column, such that in the dialysis step, the lactose, which was added in a high concentration along with the galactin-3, is moved out of the lactose column. And after dialysis, what remains is the purple target protein galactin-3. So this is the entire step of an affinity chromatography. but it also has certain drawbacks that it requires elaborate infrastructure, the whole lactose agarose column. Along with that, the lactose agarose matrix itself is very expensive. It's time consuming. From loading till the dialysis, it takes around 3 to 6 days. It requires large sample volume that is to be loaded on the column, and it's a multi-step process. Next. So in order to overcome some of these challenges of the conventional protein purification techniques, we developed a new purification technique, next, which we named it as capture and release, or in short, CARE. So in this capture and release CARE technique, the step number 1 is in the test tube, we have the entire crude. The crude has all the mixture of samples. Along with that, we have our target protein. The purple is our target protein. In the next step, we add the target capturing agent, TCA. So the red ones that is added in step number 2, the test tube, we add the TCAs. These TCAs has to be very specific to the target protein, the purple protein, such that it will only and only bind to that particular target protein and not any other protein in the mixture. So in step number 3, the TCA will bind to only the protein of interest, the purple protein, and not to any other mixture of proteins. In step number 4, the washing step will wash off all the other unwanted proteins or the other mixture of proteins. And what remains behind is that complex of TCA and the protein of interest. The next step involves the addition of the competitive ligand in a very high concentration. So the green ones are the competitive ligands, such that the role of this competitive ligand is to dissociate or separate the complex that was formed between the target protein purple and the red TCAs, such that in the test tube, what remains are the 3 different separate components dissociated, that is the TCA, target protein, and the competitive ligand. The last step involves the filtration step, which further separates the protein of interest or the target protein from the other 2 components. So this is how in this capture and release or CARE technique, we are able to purify the particular target protein, the purple protein, from the entire crude sample. And once we also purify, the next step can be protein identification to know if it is a known protein or an unknown protein. Next. So we developed this capture and release or CARE technique. Now it was the time to verify this technique. So this verification was done by one protein. We were successful in purifying one such cancer-related protein called as galactin-3. Galactin-3 is a 30 kilodalton beta-galactoside binding protein. And as we can see from this SDS page gel, that we were successful in purifying this galactin-3 using our CARE technique. And this method was further validated by purifying other known and unknown proteins as well. Next. So along with galactin-3, we also further validated this capture and release technique by targeting and purifying other proteins and glycoproteins. So in column number 1, this is a list of all the proteins and glycoproteins that we purified using CARE. And in the second column, these are the target capturing agents, TCAs, that were used, which were very specific to that particular target proteins and glycoproteins in column number 1. And in order to dissociate those proteins and glycoproteins along with the TCAs, the column number 3 is the competitive ligand that we added in a very high concentration in order to dissociate the complex and thus purify the protein or the glycoprotein. Next. So this is these are the results. This is the image of the purification of concanavalin A, con A, using CARE. So from this SDS page, we can see that lane number 2 is the commercially purified, the commercially available con A bands, and lane number 3, 4, and 5, we were successful in purifying this con A using CARE. So when we compare with our purified technique CARE with the commercially available con A, we concluded that this was this particular con A purification with care was successful. Next. Along with that, we also purified different glycoproteins using care. So in the first gel image on the left, this is the SBA, that is the soybean agglutinin, is one kind of a plant lectin, which we were successful in purifying using the care technique. In the second one, that's another glycoprotein, tarin. Tarin is one kind of anti-cancer protein, and we were also successful in purifying that particular glycoprotein tarin using care. And in gel number 3, that's the glycoprotein ovalbumin. Ovalbumin is obtained from chicken egg white. So we were also successful in purifying this particular glycoprotein using care. So So along with the development of this technique, we were also successful in purifying most of the proteins and glycoproteins using this capture and release or care technique. Next. So this particular care have several advantages. One is it is cost effective. Just like affinity chromatography, it does not require any agarose lactose column nor any antibodies.
You have 1 minute left.
Yeah, it is fast and simple. It takes like few days to, it takes like few hours to one day is enough. It requires small sample volume, requires limited lab space, and it has a specific binding. Next. So we developed, we developed a new and a convenient approach, which was like time saving, precise, cost effective, and it required less space and infrastructure. This particular care technique required only a small amount of starting material, and this is a very valuable tool to discover significantly proteins and glycoproteins. In future, this particular technique can be used to isolate or purify even the COVID-19 antibodies, or even it can be used as a detection tool to isolate the virus itself. Thank you.
All right. Are there any questions from the judges?
Well, I have several, but because I do have several, I'll defer to the other judge if he wants to begin.
Thank you. Actually, I just have a quick question. Are you going to publish some of the results based on the research you are presenting here?
Yeah, we have already one publication from this whole entire big project. It's in JBC. And yeah, and we have this project that is to be continued with more samples and go to the next level, cellular level, towards virus cells, towards cancer cells. So yeah, it's an ongoing big project.
Great. Another quick question is this KARE, capture and release. So it's a totally new idea from you guys?
Yeah.
All right. Okay. I have no further questions.
Thank you.
All right. Well, I'll start with just a financial one and then go to the technical ones. This seems to me to be highly patentable work. Do you have a patent in progress?
Yes.
Okay, good.
Yeah.
All right. So my technical questions are, first, I'm not really all that familiar with the structure or molecular weight of these proteins, but are you aware of any technical limitations, be it, is there just a range of molecular weights that this would be effective with, and or are there certain like binding sites that that your what did you call them, your the capture agents, are they only effective on certain binding sites? So are there any limitations in category of protein that you think this might not work on?
Yes, definitely. One particular drawback about this particular technique is that when we are capturing this protein with its target capturing agent, both the protein and especially the target capturing agent has to be a multivalent, so that it will form a complex. If it is a single, if it has a single binding site, and if it is not forming a complex with its binding partner, then this technique won't work. So definitely the protein and the TCA has to be a multivalent one. So this is one definite drawback of this particular purification technique.
No, I understand that, or at least I think I understood your answer, but that capture, I understand it has to be matching, but what I'm saying is, are there certain types of sites where you simply would not have the ability to have an appropriate capture agent? Maybe the binding site is so polar you can't make the capture agent release, or maybe your separation, just because the molecular weight of the protein is too high or too low. Is there any structure property limitation period?
Yeah, I mean, that is one thing which I have noticed, that even the molecular weight limitations is one factor. If one target capturing agent, for example, is really too big, and if the protein is really too small, then it becomes a little difficult during the experimentation to capture everything. So yeah, definitely for some proteins, it is a limitation.
Okay, then one final question. I really don't understand why the mannose seems to make it release. What is the chemical mechanism that causes that release? I could understand if the mannose binded, if it like kicked out the other agent.
Yeah.
You know,
Okay.
I don't understand the interactions.
Okay, so when I'm adding the competitive ligand mannose, the important point over there is I add it in a very high concentration. So because of that high concentration ability of mannose, it separates out what was bound together already, because that also mannose also has the affinity towards that particular target protein. So it will start fighting with the TCA in order to release it from the target protein. So the main factor over here comes the high concentration. Only in that high concentration mannose, it will start fighting with the TCA towards that target protein.
But I still don't understand the driving force, because the mannose isn't actually replacing the other capture agent.
It's definitely not replacing, but it is dissociating or separating both of them. That's the task, that's the aim of that step, that I want to separate both of them.
No, I understand the aim and the objective. I guess I just don't understand how it works, because if it's not replacing it, nor is it actually capturing the captured ligand, I just don't understand what physical force is replacing that interaction. Or either you have to replace the interaction with a better one, or you have to have some sort of interaction between the new agent and the old one. It just seems like, oh, well, the system is different and now my site is empty. But if it's not, if the mannose isn't interacting with either the protein or the capture agent,
yes,
I mean, it just seems sort of magical. And I guess I would like to know what interaction that concentration is causing.
Since my competitive ligand mannose is also having the affinity towards the target protein, so when it is added in high concentration, it will separate and it will also stay bound a bit towards the target protein. And that is why during the separation or the filtration process, or even the dialysis, I am trying to separate mannose or lactose from the target protein.
All right, so the image isn't showing any sort of interaction, but it actually is there. It's just a much weaker one.
Yeah, it's a weaker one.
Okay. All right. I still don't understand why, but okay. At least I understand the principle. All right.
Okay.
Thank you.
Thank you.
Well, I was going to ask the same question, but it's Dr. Hayden asked it for me. So do you have any other questions from our judges or audience members? All right. If not, that brings us to the end of this. Our third presenter withdrew from this session. So thank you both to our presenters and our judges. You are more than welcome to stay on and converse. And we want to thank everyone for coming out. And we hope to see all of you at the Graduate Research Colloquium Awards Banquet, virtual awards banquet tomorrow from 5 to 7. All participants and judges are welcome, and audience members can come and watch the award ceremony and help to recognize and honor our graduate students for this year.
Thank you.
Thank you, everyone. So Professor Sun, can you stay on a bit to help me?
Yes.
Okay, thanks. Yeah, I just wanted